Okay, hi everyone, welcome in. Thank you for coming to today's virtual seminar. I wanted to have Karen in person, but she's all the way in Washington State, so the furthest away you could possibly be. So it's my pleasure to introduce Karen. Karen and I were in graduate school at the same time. What I like most about Karen is she's someone that gets things done. So you ask her to do something and you don't have to worry about it. And for that reason, like, we loved working together on a bunch of different committees. And we still do. We still work together now. So Karen's primary interest is in medical and veterinary entomology. You also have a background in public health. And so she's worked with a bunch of different arthropods in a lot of different places. So she did grad school in A&M and then went to Penn State and is now in Washington State. And I think she'll talk more about her research, but thanks for coming, Karen, and I'll let you take over.
All right, perfect. Thanks, Chloe, for the introduction. And hi, everyone. I want to thank you for tuning into my seminar today. I really do appreciate you guys being here today and also for inviting me here to give a seminar. So a lot of the work that I'll be presenting today is some of the new stuff that I've been working on since I started here at the USDA in Pullman, Washington, in the Animal Disease Research Unit with the USDA. And a lot of my work right now focuses on different aspects of the Babesia bovis system. So since I started working at the USDA just a year and a half ago, I think it's a year and a half ago now, I've really formulated this really big and broad question for my research program, which is how do ticks utilize their environments and their hosts? And so to answer a lot of these different questions and a lot of really just much of this large question that I have, I try to combine a lot of my experiences with fieldwork, with modeling, pathogen detection, and behavioral assays to incorporate this One Health approach in understanding the ecology and the behavior of ticks and how we can use a lot of this information for tick control. So a lot of my work now focuses on the bovine babesiosis system, but I also work in other livestock tick systems that we focus on here in the unit, like theileriosis as well as anaplasmosis. And so I also have experience working on tick-borne diseases of people, and I guess also as animals, by working in the Lyme disease system as well during my postdoc. So much of the research in my lab focuses on these three major themes that you see here on the screen. And really, these themes have been inspired a lot by my previous projects as a graduate student and as a postdoc. So this includes looking at ecological factors that bring hosts and ticks together. So that's kind of a nod to my PhD, which focused on a lot of modeling of the West Nile virus system in Texas. And then I also look at motivations for ticks to choose a certain host over others and how that affects their survival. So that's taken from my postdoc. And then looking at tick interactions with other ticks, including and as well as new pathogens and invasive hosts. So looking at those tick interactions and how these different factors affect their survival as well. So that's kind of a nod to what I do currently here and what I'm trying to establish in my lab. So really all of these themes kind of put together, hopefully can ideally help us identify or develop better tick control tools to protect people and their animals from tick-borne diseases. Now going into this idea a little bit more into One Health, I think a lot of us are probably in agreement that we've seen an increase in the number of tick-borne disease cases that are reported every year. This is not only just for people, but also for animals as well. And more than likely, the emergence of these tick-borne diseases is probably related to a couple of different factors, one of them being environmental changes. So things like climate change or anthropogenic changes to the landscape. We also have host characteristics. So things like immunological factors that are associated with some hosts over others. And even human behavior, such as decision-making processes to prevent tick bites or enact tick control efforts, or understanding why people don't take those certain behaviors to protect themselves from tick bites. So my lab, as well as a lot of other different labs, both in the US and internationally, we all try to take this One Health approach because it does provide this really nice holistic view of a disease system. And so One Health really does account for the people and the animals and the environments in which ticks and these tick-borne diseases take place and persist. And more specifically, in my lab, and really in this presentation, we'll focus on things like the possibility of spillover at the wildlife-livestock interface, as well as the potential for overlapping distributions of new ticks or new pathogens. That is, what is the possibility for a new pathogen to be transmitted by native ticks in the area, or vice versa? What is the possibility for new ticks to transmit native pathogens. Now, given that there aren't a ton of really effective options for tick control, especially at the landscape level, and since we don't have a lot of vaccines against many of these tick-borne disease pathogens, this does present a very unique challenge, but also a really unique opportunity for us to use other tools, such as modeling, to create early warning systems, or for conducting acquisition and transmission studies to identify competent tick species for different pathogens, or even developing things like anti-tick bite vaccines to prevent pathogen transmission during tick feeding. So we do have a very long ways to go in terms of One Health approaches to ticks and tick-borne diseases, but we are working on it. We still have these other tools that we can use to help us prevent tick bites and tick-borne diseases that affect people and their animals. So for this presentation, I'll talk about two different projects. So one is a modeling project on wildlife hosts for cattle fever ticks. And then I'll discuss an acquisition and transmission study with Babesia bovis and the Asian longhorn tick. So for the first part of my presentation, I'll talk about some recent modeling work that's been done on the wildlife-livestock interface. And a lot of this work was conducted by my amazing postdoc, Dr. Vera Pfeiffer. And in this study, we were really interested in the possible interactions that two wildlife hosts for cattle fever ticks, so nilgai, which you can see on the left, and white-tailed deer, which you can see on the right, we're interested in seeing their interactions with cattle and the possible implications of pretty much sharing cattle fever ticks amongst all these different hosts together in the landscape. So this isn't necessarily a tick host interaction modeling project because we actually don't use any tick data. This is mostly just a purely wildlife driven project. It's still really important for us to kind of know where these wildlife hosts are possibly moving in order to understand where these ticks could also be moving through the landscape. So for this presentation, we'll focus on the cattle fever tick, and this tick has had a very long history of being found in Texas and historically in other parts of Texas. And if you are a history buff like I am, or if you're really interested in the history of medical and veterinary entomology, I really recommend looking into the history of the cattle fever tick and the cattle fever tick eradication program. It's a very fascinating program. There's a lot of history and also a lot of drama that occurs with cattle fever ticks and their policies. And I really do think that it's a very enjoyable story and really brings warmth to my heart learning about the history of the cattle fever tick eradication program and how far this program has come since it was first initiated back in the early 1900s. So going back to cattle fever ticks, we do still see cattle fever ticks in the US, mostly in Texas, and the two that we see are Rhipicephalus or Buophilus microplus, as well as Annulatus. And both of these tick species can transmit Babesia bovis and Babesia bigemina, both of which cause bovine babesiosis. Now, as a disease, bovine babesiosis is a detrimental disease to susceptible cattle populations. And surprise, a lot of our cattle populations are susceptible to bovine babesiosis, at least the cattle populations that we have here in the US. Now, if bovine babesiosis was ever introduced into the US again, it can cost the US billions of dollars annually if it goes uncontrolled. I think the current numbers are, if it goes uncontrolled in the first year of control, it would cost us about $1.9 billion in just the first year of control. So that's a lot of money. And I think that is an underestimate because that doesn't take into account the inflation that we're experiencing right now. So it could be more than $1.9 billion today. Now, as of right now, there is no vaccine to prevent transmission of bovine babesiosis. So this does unfortunately leave a lot of our cattle populations especially vulnerable to these parasites. So while bovine babesiosis was eliminated from much of the US by 1943, thanks to the cattle fever tick eradication program, we do still see cattle fever ticks from time to time, but they're mostly concentrated in South Texas in what's known as the permanent quarantine zone. So if you look at this map here on the top right, that purple area is the permanent quarantine zone, or the PQZ, and it's along the Texas-Mexico border. So the cattle fever ticks are mostly restricted to the PQZ, but sometimes we do see them outside of the PQZ and the surrounding areas and surrounding counties. And one of the major reasons for why we find these cattle fever ticks inside and even outside the permanent quarantine zone is probably due to wildlife hosts. They act as this blood meal to sustain these tick populations, but these wildlife hosts also serve as literal vehicles, kind of shuttling these ticks throughout the different landscapes. Now, cattle fever ticks are one-host ticks, meaning that they will feed on one host for the entirety of its life cycle. It's kind of cute in a way. They find their host, and it's their host for life. So they get on the host and they feed as just tiny little larvae, and then they'll feed and they'll molt on the host into a nymph. The nymphs feed on the host, and again, they'll molt into an adult, and then the adults feed, and then the adults will mate, feed, and then the females will drop off and lay the eggs in the environment. So they spend most of their time actually on the host. Now, for cattle fever ticks, they are known to feed on cattle. They really love feeding on cattle, but they also are known to feed on other wildlife species. And within the cattle fever tick system, at least the system we see in Texas, white-tailed deer and nilgai are considered important wildlife hosts that are capable of sustaining these tick populations in the wild, as well as in the absence of cattle. And I say in the absence of cattle because these wildlife hosts actually render pasture vacation pretty much useless in this case. So pasture vacation is actually a technique that was used during the eradication program, still used today, that is used to basically starve out tick populations in an infested pasture. So what you do is basically if you have an infested pasture and you have your cattle there, you basically clean the cattle, move them to a clean pasture. And then basically for the infested pasture, you just kind of let it sit for about 9 to 12 months for those ticks to basically starve. But of course, wildlife are like, oh, great, this is an open pasture. I'm going to go in there and I'm going to eat the grass and do whatever they want to do in those pastures. So of course, those ticks can feed on those wildlife hosts, sustain the tick populations. And so you have to wait even longer for those ticks to starve out. So essentially, these wildlife hosts can render pasture vacation pretty much useless at this point. But that's not one of the only reasons why we are concerned about these wildlife hosts. There is very large concern because these animals can travel very far distances and they have very large home ranges, some of which you can see some of those numbers here that I pulled from the literature. So really taking into account the fact that cattle fever ticks are one-host ticks and they take about 3 weeks to complete their life cycle, we can kind of think of how far these animals can travel in those 3 weeks and dropping those ticks along the way, along their routes. So white-tailed deer are considered this native host for cattle fever tick populations, and they have a very long history with cattle fever ticks. Back when the cattle fever tick eradication program was at its peak, white-tailed deer were one of the hosts that were targeted as one of the hosts that were sustaining the tick population. So they've had a very long link with cattle fever ticks in the past. But we also consider this a native host in that its natural range is found throughout much of southeastern U.S., including much of South Texas. Now, we've probably all seen white-tailed deer in our life, probably in our backyards, our front yards, maybe even around campus, around town. They're a rather ubiquitous animal that we see throughout much of the U.S., but back in the early 1900s, this actually wasn't always the case. We actually see that white-tailed deer populations hit an all-time low. However, we do see quickly after people realize that they hit an all-time low that white-tailed deer experienced this huge population boom due to a couple of different events, like the eradication of screwworm. We also saw deer restocking events for conservation efforts. But we also saw market hunting elimination for deer and even economic opportunities that arose through game ranching and hunting operations to breed these deer for hunting purposes. So, of course, this led to a really large rise in white-tailed deer populations, especially in Texas. And as someone who was born and raised in Texas, I can say that Texas really does love their hunting activities, and that includes hunting deer. And so while we saw this rise in white-tailed deer populations, we also see a rise in possible cattle fever tick hosts that these ticks can now feed on. So I do tend to dunk on deer quite a bit. I do love deer. I study them for quite some time already, but they aren't the only ones to blame when it comes to supporting cattle fever ticks. We actually have another host in the landscape in South Texas that can also serve as a host to support these cattle fever tick populations, and that's the nilgai. Now, unlike white-tailed deer, which are naturally found throughout much of the southeast U.S., nilgai are actually relatively new ungulates that were introduced into Texas back in the 1920s, mostly for hunting purposes. And since they were actually introduced in the 1920s, nilgai are actually considered established throughout much of the southern parts of Texas. And by established, I mean there are wild populations of nilgai throughout much of South Texas. Now, this isn't super surprising, given that nilgai are considered this generalist animal. They can adapt to many different types of habitats, and normal game fencing actually can't keep them contained. And so I am about I think someone might be on. So I am actually about 5'3", 5'4" on a really, really good day. And I only measure up to about the shoulder height of a fully grown adult nilgai. So these are very large animals. So normal game fencing that can normally keep white-tailed deer in or out, depending on which side of the fence you're on, normal game fencing can't keep these animals contained. And really, that's how a lot of these nilgai escaped. And that's how they've established throughout different parts of southern Texas. And so given the possible ranges that nilgai and white-tailed deer can travel, it's really important that we start to understand where these animals can travel, where they can potentially inhabit and potentially come in contact with cattle. And that kind of brings us to this really major question for this part of the study, which is, what is the risk of either or both of these wildlife hosts coming in contact with livestock in the southern US? And in the study, we created species distribution models for each of our wildlife hosts. So one for white-tailed deer, one for nilgai. And then what we'll do is we'll calculate where these two species overlap with cattle, and then measure how much contact will be increased when both of these animals are present in the landscape. And so hopefully this information can give us some indication of possible interactions between livestock and cattle fever ticks if these wildlife hosts happen to bring any of these ticks with them. So the study extent for this research focused on Texas, but we did add a couple of surrounding states like Kansas and Arkansas, Oklahoma, and Louisiana. And we gathered all of our data from a variety of different sources. So for our occurrence data for white-tailed deer and nilgai, we grabbed occurrence data from the Global Biodiversity Information Facility, or GBIF. And so we have white-tailed deer occurrence in our study extent here in panel A in the top left. And we also have nilgai occurrence in our study extent, which you can see in panel C on the right-hand side. But as you can see, we actually don't have too many occurrences of nilgai, or at least reports of nilgai sightings in the US. And so what we did was we actually built our nilgai species distribution model based on the distribution of nilgai in their homeland range in India, which you can see in panel B in the middle. And then we projected it to our study extent in the southern US. Now in panels D and E, these two layers are very important because they'll calculate cattle density for us. And so we have cattle population data from the USDA National Agricultural Statistics Survey, and that tells us basically the cattle population per county. And then finally in panel E in the bottom right, we have cropland data from the USDA that's been reclassified into suitable versus non-suitable lands to raise cattle. So both of these panels for D and E were used to calculate this other layer, cattle density. And this is actually a very important layer because it helps us calculate the risk of cattle contact with white-tailed deer, nilgai, or both of these animals in the landscape. So for cattle density, we distributed the cattle inventory per county, which again from panel D, and then we distributed that across the area of land suitable for cattle, again from the image here in panel E. So we have three broad categories in terms of our explanatory variables that we wanted to add into our model. So that included different variables from the environment. We also had some topographic variables and land use variables. And all of these explanatory variables went into a random forest model, which is a type of machine learning modeling method. And the random forest models will help us identify the variables that are most likely to contribute to the distribution of white-tailed deer and nilgai that we might see in our study extent. So to conduct this work, we first calculated our SDMs for white-tailed deer, and then we calculated the SDMs for nilgai separately. And then what we'll do later in a few more slides is we'll calculate a combined exposure risk, putting those two layers together and then with cattle. So first, for our white-tailed deer models, we first identified our top contributing variables and then built our SDMs based on those variables and then projected it for the southern US and our study extent, and then more specifically for the permanent quarantine zone down in South Texas. And then we calculated this additional layer that tells us the probability of contact between cattle and white-tailed deer using the equation that you see at the bottom of the screen. So to develop our white-tailed deer species distribution models, we found that elevation and winter precipitation were our top contributing variables to that distribution model. And when we created this model and then projected it to the southern US, we see that white-tailed deer presence was likely to taper off in the arid west, with the darker colors representing lower habitat suitability and the lighter colors representing higher habitat suitability. So this is kind of the scale that we're working with for the rest for a lot of our maps that I'll be presenting today. So lighter colors mean higher habitat suitability, and dark colors mean lower habitat suitability. Now, when we zoom in on the permanent quarantine zone, we see that white-tailed deer occurrence is going to be, is expected to be higher, at least, in the eastern counties of the Texas coastal plain. So if you kind of look here in the top right of the map with a lot of the yellow colors, they're expected to be the highest population density in that area. But we also see along the permanent quarantine zone, there is some, still some pretty good habitat suitability for white-tailed deer. Now, in terms of cattle contact with white-tailed deer, it seems to be the highest in Texas and Oklahoma compared to the rest of the spatial extent that we have. And so for this type of map, basically this color ramp is telling us, you know, the lighter colors represent the number of cattle heads that are potentially coming in contact with deer, and then with the darker colors, that's less cattle heads coming in contact with deer. So for the nilgai species distribution models, we basically have the same exact procedures, but we did have that one extra step. So remember how I said that there were barely any nilgai occurrences in the US? So we had to go back to the distribution of nilgai in India and build our SDMs based on that. So we identified our contributing variables based on the India distribution, projected that model for India, and then projected it for the southern US and the permanent quarantine zone in Texas. And just like our white-tailed deer models, we calculated the probability of contact between cattle and nilgai using this equation that you see at the bottom of the screen. Similar to white-tailed deer, elevation was yet another top contributing variable to the nilgai model. But we also see that nilgai were heavily affected by minimum temperature in the coldest quarter. So I know I said earlier that nilgai are very adaptable to many different environments, but I meant many different warm environments. So nilgai are actually susceptible to freezing temperatures. And so in, I think, 2021, nilgai, there are actually a very large number of nilgai mortalities that were reported in 2021 after the huge freeze that happened back in February of 2021. Now, based on the original distribution of nilgai in India, we see that nilgai will likely be concentrated in southern Texas, as well as just a little bit in Louisiana, just at the very tip of southern Louisiana. And when we zoom into permanent quarantine zone in southern Texas, we expect to see our nilgai in counties along the Texas-Mexico border. So lots of really nice habitat for the nilgai there. But we also see some possible habitat for nilgai along the Gulf Coast as well. And we also expect cattle contact with nilgai to be the highest in Texas. And this kind of checks out in a way because that's kind of where the nilgai currently are. And so we know the potential contact risk of cattle with white-tailed deer, and we also know the cattle contact with nilgai separately. But now we want to know how much are cattle at risk of coming in contact with either of these hosts, with both of these hosts in the landscape together, which that can be calculated using the first equation that you see on the screen. And then finally, we want to see how much contact risk is increased when we add nilgai to the landscape. And so remember how I said white-tailed deer are considered these native hosts. So they provide this baseline risk of cattle coming into contact with any wildlife host. And so now we want to see how much that risk is increased when we add nilgai to the landscape. And we can calculate that percent increase using the second equation that you see on the screen. So in doing this, we found that white-tailed deer or nilgai are likely to come in contact with cattle in the eastern parts of our study extent, including eastern Texas and Oklahoma, and actually much of Arkansas and Louisiana. But when we calculate the percent increases, we see that Texas had the highest percent increases in cattle exposure to either of these wildlife hosts, with an almost 20% increase. And so basically what this is telling us is that compared to just having white-tailed deer potentially coming in contact with cattle, when we add nilgai to the landscape, we see an almost 20% increase in the number of cattle coming in contact with any wildlife, or I guess either of these wildlife hosts, when both of the animals are present. And so when you kind of think about it, Texas has a lot of cattle. And so they have millions of heads of cattle in the state. And so just a 20% increase in contact it's quite a lot, probably hundreds of thousands of cattle heads coming in contact with either of these hosts. Now looking in the permanent quarantine zone, Zapata County, which you can see as that teal area that's circled on the map, they are likely to experience the greatest percent increased risk of cattle contact with wildlife, with almost a 48% increase. So wildlife hosts are really some of the major reasons why we don't have complete eradication of cattle fever ticks. And they're also likely vehicles for ticks to travel outside of the permanent quarantine zone. And so really these models just confirm that having more than one wildlife host will increase that exposure risk of cattle to these hosts. And thusly, that could potentially increase the chances of cattle fever ticks entering these areas where cattle are if these wildlife happen to carry those ticks with them. And furthermore, we do expect these interactions between cattle and wildlife to be the highest in South Texas, with the potential for range expansion of wildlife hosts going even further north in the face of climate change. So remember how I said nilgai are very adaptable to warm climates? If climate change renders our world warmer, that could mean that the nilgai can also expand their range and go further north. So this study is actually currently in review in the Journal of Medical Entomology. So hopefully we get an acceptance soon for this paper, and we really look forward to having the paper published pretty soon. Okay, so this work really provides kind of the first set of models that show us how wildlife hosts of cattle fever ticks can overlap, and it shows us contact between cattle populations and how this may affect tick transfer amongst these different animals. Now, one thing that I think our current model is missing is really how these animals can potentially move across the landscape. Currently, our models just show us the static view of where these animals can potentially inhabit, but now we actually want to see, like, how are these animals actually getting to these locations, or can they even get to those locations? And so one of our next steps for this project is looking at barriers to movement of these animals and how this might affect potential establishment of these animals in new areas. So we're going to be using a program known as Omniscape, and this applies a circuit theory through a landscape lens. So you kind of have this moving window with a specified radius that you give it, and basically what the program will tell us is if and how barriers can affect successful animal movement and establishment. And so basically, kind of thinking back to circuit theory and kind of our physics class, we'll add in what's known as a resistance layer. And this is defined as any barrier or any traversal cost that an animal has to put out in order to cross the landscape. So in this resistance layer, it can be anything like highways, it can be high fences, or even, large bodies of water that these animals can't really cross. And so that would be a very high cost for those animals to cross these different barriers. So this is considered a very high resistance to animal movement. And so within this moving window, Omniscape will calculate this flow of quote unquote current, or in this case, animal movement, based on how much resistance is placed in that landscape. So while our current model tells us where the animals are likely to be found in the landscape just based on their habitat preferences, landscape connectivity can tell us the probability of these animals to travel across the landscape, or even more importantly, why some of these animals may not establish in a new area. Now, another project that we're looking into is doing some forecasting of tick populations based on different weather patterns. So while I wish I could just look into a crystal ball, shake it, and ask it when and where are those cattle fever ticks going to be active. We don't really have a tool like that, but we do have something that's sort of like that, and models can help us get as close as we can. So given that the cattle fever tick system has actually been in the US since around the 1600s, and the control program was established in the early 1900s, there's actually a lot of historical data on tick infestations along the Texas-Mexico border. So hopefully using some of those historical data that USDA APHIS has collected, we can apply time series models, and we can not only identify patterns in cattle fever tick populations based on weather and time, but we can hopefully look to the future and forecast when and where we're likely to see these ticks in the future. And really the goal at the end of this kind of forecasting work is, I envision a tool that stakeholders can use to track ticks and weather in real time, so they can help them with some decision-making processes on whether they want to initiate prevention and control efforts, and hopefully, save some time, money, and effort for some of our stakeholders when it comes to controlling for these ticks. So those are two kind of the biggest projects that I have happening currently. We really can't wait to continue this work and hopefully have this work published in the future as well. All right, friends, we're actually going to take a hard right. We're going to shift gears really quickly into another type of study that's currently ongoing in my lab. It's a very different experiment compared to the modeling stuff. This is an acquisition and transmission experiment. And so while we've talked about, a tick that's been in the US for quite some time, we can talk a little bit about a tick that's actually newer to us in the US, and that is the Asian longhorn tick. So for this part of the presentation, We're trying to see if Asian longhorn ticks are capable of transmitting Babesia bovis. So for this experiment, we do have two main characters that we're interested in. The first of which is the pathogen itself, so that is Babesia bovis. So for the Babesia bovis life cycle, there are two main parts to it. The first part is the sexual stage of the parasite, and that takes place in the body of the tick. And then the second stage is the asexual stage, which happens in the bovid host. So for today's talk, we're mostly just interested in what's happening in the tick. So we'll just be focusing on the sexual stage of Babesia bovis, again, which takes place in the tick. So for the sexual stage of Babesia bovis, that's initiated when a tick actually ingests the gametocytes of the parasite during that blood meal on the mammalian host. And then following ingestion, the gametocytes differentiate into male and female gametes within the tick's midgut. And then those gametes fuse together to form a zygote. The zygote will then develop into an ookinete that then migrates to the different various organs within the tick. So Babesia bovis is actually transmitted transovarially. So the adult female will actually pick up the pathogen, but it's actually the larval stage of the second generation that ends up transmitting the pathogen to a new host. So knowing that it's a transovarially transmitted pathogen, the kinase must actually have to gain access to the ovaries of the female in order to be transmitted to the eggs, which then get transmitted to the larval stage. So to do that, the kinases have to escape the midgut, make their way to the hemolymph highway, as I like to call it, and then they use the hemolymph and travel and invade the ovaries and then infect the eggs. And then eventually the parasite will make its way to the salivary glands of the larval tick, and it's during this migration that the kinases transform into sporozoites in the salivary glands, which are then transmitted by the tick to a new vertebrate host during a blood meal. And then that kicks off the asexual cycle that occurs in the bovid host. Now, the other main character we're interested in is the Asian longhorn tick, or Haemaphysalis longicornis. So unlike cattle fever ticks, which are one-host ticks, cattle fever ticks, the Asian longhorn tick is a three-host tick, meaning that they take a blood meal from 3 different hosts during its lifetime. Now, the really fun part of the tick, at least I think it's really fun, is that the tick that we have the strain that we have in the US is considered parthenogenetic, meaning that females can reproduce essentially asexually and essentially produce clones without a male. So in other words, all you really need is just one female to start a whole new colony in a new location, which really makes it a great case study for an invasive tick, but also an invasive animal in general. Now, as far as animal hosts for this tick, we found this tick on many different types of animals, including livestock, domestic animals, as well as wildlife. And because of its parthenogenetic nature, this tick can reproduce somewhat quickly. And if you find high abundances on this tick on an animal, that can cause a lot of stress on an animal, which in turn reduces a lot of the growth and production potential for that animal as well. And at high enough abundances on a single animal, this tick can pretty much exsanguinate an animal. So the ticks basically just drink all of the blood in that animal, kind of like tiny little vampires. And this has been reported, I think, at least in a couple of calves and perhaps, I think, a fawn as well. Now, as far as transmitting pathogens, a lot of research is still ongoing, but Theileria orientalis has been detected in this tick in the field, and it's also been confirmed in the laboratory that this tick can transmit Theileria orientalis akita. The pathogen that causes Rocky Mountain spotted fever, or Rickettsia rickettsii, that can also be transmitted by this tick, but this has only been tested in the laboratory, and as far as I know, I don't believe Rickettsia rickettsii has been detected in this tick, at least not yet. Now, more recently, a couple more papers have come out saying that the tick can also transmit Powassan virus as well as Heartland virus in the lab. But again, that's mostly been tested in the laboratory, and I don't think that it's been detected in the field just yet. Now, when I say this tick is new on the block, it is really new. The first report of this tick was actually back in 2017, at least the tick found in the US. And the first report was made in 2017 in New Jersey, where you guys are, where Rutgers is. And this was reported by Rainey et al. However, after this report came out, some folks looked at some historical tick specimens, and it turns out that this tick has likely been in the US since about 2010. So this tick has had more than a decade to pretty much establish itself in the US. So since this report, the tick has quickly spread throughout much of the eastern United States, with reports from at least 17 states, although I think this number might have changed a little bit more recently. It might be 18 or 19 states now. So looking at this map that I grabbed from APHIS, we can see that this tick is mostly found along the East Coast, specifically in states in the mid-Atlantic and going into the Southeast United States. But there are reports going as far west as Missouri and Arkansas. But what we actually don't see, though, is any Longicornis that have made its way to Texas. Well, in my opinion, I think it's, we just haven't seen it yet. As with all things, I do think it's only a matter of time before this tick actually does make its way to Texas. It's already been detected in neighboring states, such as Arkansas already, but now we're kind of just wondering when it's going to make its way to Texas. Now, according to a species distribution modeling paper that was published by Raghavan et al., we can see that South Texas can be a pretty nice place for these ticks to survive. And some of this area does seem to overlap with where we've seen cattle fever ticks, especially in South Texas, but also historical detections of Babesia bovis throughout much of the Southeast United States. So just to give a little bit of background on this paper and how to interpret it, they tested out 3 different models using maxent modeling methods. And so the more models that are in agreement saying that this tick is likely to inhabit a certain area, and the higher the probability that that may be true. So down here in South Texas, it seems like maybe 1 or 2 models are in agreement with each other saying that this tick could be in Texas. So as long as there's one model who could predict that this tick can make its way to Texas and survive there, that's enough for me to be a little bit worried about the cattle populations in Texas. So overall, this could be pretty bad news for our stakeholders and our landowners. If Babesia bovis does come in contact with the Asian longhorn tick and the tick is able to acquire and transmit that pathogen, that can be really bad news for our livestock owners and the cattle industry, especially given the tick's parthenogenetic nature, but also because a lot of our cattle populations in the US are considered susceptible to Babesia bovis. But so far, there haven't been any studies looking into the transmission potential of Babesia bovis by Longicornis. So that's kind of our main objective for the study. It's whether or not Longicornis can acquire and transmit Babesia bovis. And if they can, what are the life stages that are responsible for transmitting this pathogen? Okay, so I'm not going to lie, the experimental plan is probably going to get a little chaotic, but I promise that I will try my best to walk everyone through all these different experimental steps, just one step at a time. So we have 2 main components to the experiment, and those you can see them here on the screen. The first phase is the acquisition stage, and this is where ticks acquire the pathogen through an infected animal. So we basically have to inoculate a calf, add the ticks onto that infected calf, and then the ticks, after they're done feeding, should presumably be infected with Babesia bovis. And then we have the second stage, which is the transmission stage, where we see if the ticks can transmit the pathogen to a new host. So we add the presumably infected ticks from our last step, put it onto a new calf, and then we wait and see if the calf develops any signs of Babesia infection. So at the heart of this experiment, this is pretty much what we're doing with this project. Where it gets a little crazy is when we talk about the different transmission routes for Babesia bovis. So for this experiment, we are testing whether Babesia bovis is transmitted via transstadial or transovarial transmission. Now we know Babesia bovis is transmitted transovarially via Rhipicephalus or Buophalus microplus, but perhaps it might take a different route in Longicornis, which is why we're testing both routes. So for transstadial transmission, we are seeing whether the pathogen is transmitted from one life stage to the next. And so in this experiment, we're going to see if the nymphs can acquire or pick up the pathogen. We'll allow those nymphs to molt, and then we'll put the adults, the females, onto a naive splenectomized calf. And then for transovarial transmission, we are seeing whether the pathogen is transmitted from one generation to the next, so from mother to offspring. And there's just a couple more steps with transovarial transmission, but basically what we're going to do is we're going to have the adult stage acquire the pathogen, and then seeing if the second generation of ticks, whether it's the larval, the nymphal, or the adult stages, see if any of those stages can transmit Babesia bovis to a calf. Okay, so the first step in our experiment is the acquisition stage, so the acquisition feeding here. And this is basically where we infect the ticks with Babesia bovis. So in this step, we have to synchronize our tick feeding and repletion with peak parasitemia of Babesia bovis in an infected splenectomized calf. That is, we want to make sure those ticks are fully replete during peak parasitemia of Babesia bovis in the calf so that we maximize our chances of having infected ticks for our transmission segment. So we infected our calves with a stabolate of Babesia bovis, and then we added our Longicornis nymphs and adults to a calf, which you can see here on the right-hand side of the figure. And then in the schematic on the left, we are using the MicroPlus system as our positive control for our experiment. So we don't have to wonder if something went wrong during the inoculation phase. So if during the transmission, if our MicroPlus calf never falls sick, then we know that something went wrong in our experiment. And that's really kind of the purpose of your positive control in an experiment in general. So we added our larval MicroPlus ticks to a calf, because remember, these are one-host ticks, so we actually have to rear the larval ticks before we inoculate the calf. So by the time those MicroPlus are about between like the nymphal and adult stage, we inoculate the calf, and then by the time the calf reaches peak parasitemia, the adults for MicroPlus should be reaching full repletion. Okay, so here's kind of the timeline of the acquisition feeding in case you were interested. So we actually started this experiment back in January, which is kind of crazy to think about. And you'll see why in a couple slides, why I think that's why the timeline is kind of crazy. But basically, we inoculated both of our acquisition calves on January 30th. That was kind of the big day of inoculation. And then by February 7th, those calves were PCR positive for Babesia bovis. And just a few days later, the calves were showing severe signs of bovine babesiosis, so they were terminated for this experiment. And in total, this phase was about 11 days total between inoculation to termination. So it's a very rapid process that this disease takes place. So to test whether the ticks acquired Babesia, we took a subset of MicroPlus and Longicornis that fed on the calves, and we processed them in a couple of different ways while allowing the rest of the ticks to molt or lay eggs for our next step in the transmission experiment. So for the Longicornis adults, we dissected the midguts and the ovaries from the ticks at 5 different time points, so 4, 6, 8, 10, and 12 days post-repletion. And we kind of did it this way so that we can kind of see the progression of Babesia through the body of Longicornis over time. And so after we dissected out the midguts and the ovaries, we then tested them for Babesia bovis using a nested PCR with kinase-specific protein primers. And since the nymphs were so tiny, it would actually take forever for us to dissect out all the different parts that we collected for the adults. They're like, no, let's not do that. So instead, we actually just macerated the nymphs whole using those same different time points and then using the same protocol to test them for Babesia bovis. Now, when we tested Longicornis ovaries and midguts, we found that the percent of positive ovaries and midguts both increased as the number of days post-repletion and removal from the animal progressed. And most notably, as you can see in the graph, 12 days after the ticks were removed, we see a very high percentage of samples that were positive for Babesia bovis. And for the nymphs, the timeline was a little bit less consistent compared to what we found for the adults. But we do see that a very high percentage of positive nymphs were found 6 days after the ticks were fully fed. And now remember that Babesia bovis is typically transmitted transovarially to the second generation. So those kinases have to make their way to the ovaries, they have to make their way to the eggs, and then eventually into the salivary glands for the next generation of larvae. So we were interested to see if the eggs were going to be positive for Babesia bovis. So we took egg batches from MicroPlus and from Longicornis. We ground them up and we tested them in our nested PCR in triplicate. And while all three of our MicroPlus egg batches were positive, none of the egg batches for Longicornis were actually positive. So kind of conflicting results when it comes to evidence for Longicornis to acquire Babesia bovis. But I think the real test here is whether or not these ticks can transmit Babesia bovis to a new host. So after about 2 months of very patiently waiting for our ticks to complete their molting and their egg laying process, we were ready to start our transmission experiment. And so just as a reminder, we are testing both transstadial and transovarial transmission for Longicornis, and we are using MicroPlus as our positive control for our transovarial transmission. So for Longicornis transstadial transmission, we're going to be putting the adults that molted from the nymphal stage onto our transmission calf. Then for microplus and the longicornis transovarial transmission, we'll put larvae onto the calves. So in this phase, we had 3 calves to test for the microplus transovarial transmission, the longicornis transstadial transmission, and then the first part of our longicornis transovarial transmission. So we basically apply the ticks to their respective calves, and then we basically just wait for infection to develop in the calves if they have any. So for microplus, we kind of know this timeline pretty well. And so we basically are waiting about 11 or so days for signs to start showing up with the calf. Now for longicornis, we are not really sure the timeline. That's really what we're trying to figure out. We're trying to figure out this timeline. So we allow 3 times the normal amount of time for the calf to develop signs, and then add a couple more weeks just for good measure, and that equates to about 45 days total. So we're waiting about 45 days after we put longicornis onto these calves to see if they develop any signs. So for microplus, we placed 1 gram of larvae, which equates to about 20,000 ticks onto a calf. And the calf was PCR positive just about a week later, and then was terminated by May 15th, or I guess just a few days after the PCR positive sign. And so we terminated this experiment due to severe signs showing in this calf. And then we took a subset of partially fed microplus larvae and we tested them for Babesia bovis with 4% of those ticks testing positive. And really at this stage, the microplus positive control experiment is actually completed because the nymphal and the adult stages of microplus actually don't transmit Babesia bovis. So we're pretty much done with microplus, and we're not going to see them for the rest of this experiment. But seeing how this calf actually did develop severe signs of Babesia infection, we know that our experiment, at least our acquisition experiment, worked. So for the longicornis transstadial transmission, we placed adults onto a calf, and for the full 45 days, that calf stayed healthy. It never developed any signs of Babesia infection, and it was PCR negative for the whole 45 days. And just like microplus, we did take a subset of partially fed longicornis. And so for this, we took the adults and dissected out their salivary glands. And we did this because we suspect that if a parasite was found in the salivary glands during feeding, then the parasite was able to successfully escape the MIG gut, make it to the molt, and then arrived at the salivary gland and was getting ready to be transmitted to the host. When we tested the salivary glands, only 1%, or really 1 set of salivary glands, were positive for Babesia bovis. And so at this stage, we actually completed our transstadial experiment as well. And all that's really left is this transovarial experiment. So for the longicornis transovarial transmission, we placed 1 gram of larvae onto a calf back in April. And like our transstadial transmission calf, this calf remained PCR negative and never developed any signs of Babesia infection. And so this experiment was terminated 45 days later. Then we took a subset of those partially fed larvae, tested them for Babesia bovis, and 2% of them, or 2 sets of larvae, were positive for Babesia bovis. And so the rest of the ticks that were feeding on the calf, they were allowed to feed to repletion, and then we allowed them to molt to the nymphs for the next steps of our experiment. And then we put 750 nymphs onto a calf. And again, I'm sounding like a broken record, but the calf was PCR negative and never developed any signs of Babesia infection. So this experiment was terminated on day 45. We took a subset of the partially fed nymphs and tested them for Babesia, and we did not detect any positive nymphs at this point. And the rest of the nymphs were allowed to feed to repletion and then allowed to molt to the adult stage. And so we put 500 adults onto a calf, and just like our other calves, again, sounding like a broken record, this calf did not test positive for Babesia and never showed signs of infection. And so technically this part of the study is still ongoing, but we are currently on day 43 with this calf. So unless that calf ends up being PCR positive or starts showing signs within the next 2 days, the experiment will terminate on October 22nd, which will be day 45. I think that's a Sunday. So we took a subset of the partially fed adults and dissected out the salivary glands and then tested them for Babesia. And we did not detect any Babesia in the salivary glands from these adult ticks. And with that, we are basically done with the transovarial experiment, and really this experiment as a whole. Well, sort of. We have 2 days left, but I'm considering it done. So after about 11 months of conducting this experiment, which really just consisted of a lot of very patiently waiting for our ticks, we can finally make some conclusions from this experiment. And so from this project, we can conclude that there is some evidence of Babesia bovis acquisition by Longhorned, but as you saw, there seems to be little to no evidence of transstadial transmission or transovarial transmission of Babesia bovis by Longhorned. Okay, so remember how I said bovine babesiosis is actually caused by 2 different Babesia species. So our immediate next step after we're done with this project is to do this entire experiment all over again, this time with Babesia bigemina. So yet another 11 months of my life will be taken up by this project. Again, mostly waiting, but still 11 months nonetheless. So much of this study actually took place in tandem with another project that's led by Tammy Johnson and Pia Olofsson in Texas, and they're testing whether nilgai are reservoirs for Babesia. So this really ties into the first part of this talk, where I talked about how deer and nilgai are hosts for ticks. But eventually, we do want to model Babesia in the US. So it's really important that we know the reservoir status of these 2 wildlife hosts to really accurately model the potential spread of Babesia via these animals. Now, we know that deer are actually not reservoirs for Babesia. That's been shown in the literature already. But now we want to know if nilgai can serve as reservoirs, because technically nilgai are bovid animals, so they're very closely related to cattle. So if they are reservoirs, that could have some effect on how we model Babesia in the future. So in the very near future, we will be publishing this study as well as the nilgai and Babesia bovis infection study that Tammy and Pia are leading. And then we'll all together embark on a new adventure with Babesia bigemina. So lots of really cool stuff coming out soon, or at least I think it's really cool, but I'm also really biased towards the work that I do. So definitely keep an eye out for those different publications, hopefully coming out in the next year or so. So again, here's just another snapshot of my research program. And overall, my lab, again, really tries to focus on that One Health approach to investigate ticks and tick-borne disease systems. We do primarily focus on the actual tick itself and the vector itself, but we try and research how the tick interacts with the different components of the One Health paradigm. So seeing how it interacts with new pathogens or how it could potentially spread through a range based on different host presence. And altogether, the research in my lab hopes to inform and help prioritize these tick control efforts and perhaps even policy to stop ticks in their tracks, basically. So overall, by understanding how arthropods utilize their environments and their hosts, we can build this really nice tick prevention and control plan, or hopefully tools that can be implemented earlier or more effectively to prevent disease in different populations. So as you can tell, it really does take a village to get a lot of this work done, especially the acquisition and transmission experiment. So huge thank you to everyone listed on the screen for their contributions to this project, however little or however large that contribution may be. And of course, thank you to Rutgers Entomology for inviting me to give a seminar today. And I do want to thank y'all for joining me today as well and listening to my presentation. So if you want to reach out, feel free to contact me using the information on my screen. Otherwise, I can go ahead and take any questions now. Thank you.
Yeah.